Endocrinology, cell surface receptor, extracellular receptors. Majority of signaling molecules that are either hydrophilic or too big to rapidly cross plasma membrane bind to cell surface receptor and transduce signal through second messenger. Cell surface receptor classification. According to mechanism of action, cell surface receptors are receptors coupled directly to effector molecules. They don't need second messengers, ligand gated ion channels, enzyme linked or catalytic receptors. Receptors coupled via G protein to membrane associated enzymes to produce second messengers. Receptor coupled to adenyl, adenyl late cyclase. Receptor coupled to phosphatidyl inositol hydrolysis. Mechanism of action of second messenger. Mechanism of action through intracellular receptor cytosolic receptor. Through intracellular receptor nuclear receptors. The ligand receptor complex is a signal for the group 1 hormones. The lipophilic group 1 hormones diffuse through the plasma membrane of all cells but only encounter their specific high affinity intracellular receptors in the target cell. These receptors can be located in the cytoplasm or in the nucleus of the target cell. The hormone receptor complex first undergoes an activation reaction. Group 1 hormone receptor activation occurs by at least two mechanisms. Glucocorticoid diffuse across a plasma membrane and encounter their cognate receptor in the cytoplasm of the target cell. Ligand receptor binding results in a conformational change in the receptor leading to dissociation of the heat shock protein 90. This is a diagram. Then this steps appears to be necessary for subsequent nuclear localization of the glucocorticoid receptors. This receptor also contains a nuclear localization sequence that is now free to exist in the translocation from a cytoplasm to the nucleus. The activated receptor moves into the nucleus and binds with a high affinity to the specific DNA sequence called the hormone response element HRE. The DNA bound liganded receptor serves as a high affinity binding site for one or more coactivator proteins, and accelerated gene transcription typically occur. This is a diagram. After the hormone binding, the receptor releases its cytoplasmic binding protein and the DNA binding domain is thereby exposed. Together with the bound hormone, the activated receptor is thus able to bind the hormone responsive element HRE. Group 1 hormones, by contrast, certain hormones such as the thyroid hormones and retinoid diffuse from the extracellular fluid across the plasma membrane and go directly into the nucleus. In this case, the cognate receptor is already bound to the HRE. However, this DNA-bound receptor fails to activate transcription because it exists in a complex with a core pressor. Indeed, the receptor core pressor complex serves as an active repressor of the gene transcription. This is a diagram. Group 1 hormone association of ligand with these receptors results in dissociation of the core pressor. The liganded receptor is now capable of binding one or more coactivators with high affinity, resulting in the activation of gene transcription. Group 1 hormones, by selectively affecting gene transcription and the consequent production of the appropriate target messenger RNA, the amount of specific protein are changed and metabolic processes are influenced. The influence of each of these hormones is quite specific. Generally, a given hormone directly affects greater than 1% of these genes, mRNA, or protein in the target cell. Sometimes only a few are affected.